Hello, I am Myrto, and on behalf of our all co-authors, I would like to present to you our poster on imagining our hands, feet, and our whole body. When we are talking about imagining, we specifically refer to motor imagery, which is imagining of an action without the physical execution of the movement. Our study aims to explore whether there are differences between hands, feet, and a whole body in more implicit and more explicit motor imagery tasks. We focus our attention to more implicit and more explicit measures, as associations have been reported between them in the literature. In addition, since the majority of the motor imagery research has focused on hands and our whole body and less often to our lower body parts, such as our feet, we aim to compare those three body districts as there are anatomical, functional, and sensory differences between them. For that reason, we recruited 60 individuals who completed a series of online experimental tasks. We first measured motor imagery through an implicit task, that is, an implicit association test for hands, feet, and the whole body. Then we measured motor imagery through an explicit task, that is, a metamotochronometry task for hands, feet, and the whole body. And finally, to control for the effect of visual imagery, we administered a vividness of visual imagery questionnaire for hands, feet, and the whole body. In terms of results, we did not find the main effect of body district on the more implicit task. However, participants did indicate that they associate their hands with a motor imagery attribute, whereas no associations were found for the feet and the whole body. Now, in terms of the visual imagery, we found a main effect of the body district, with participants reporting a clearer image of their whole body than their hands and their feet. Whereas for the motor imagery explicit task, we first identified that participants' motor imagery time and motor imagery execution were correlated. Thus, isochrony was present in all body districts. However, this strength of isochrony when compared using Fisher's R2Z transformation was not significantly different across the three body districts. So what do those results mean? Those results suggest that motor imagery does not vary between body districts and across tasks. They also suggest that those findings are not due to visual imagery differences, as we did find some differences across the body districts on the vividness of visual imagery. Now, the different results across visual imagery and motor imagery could be related to the imagery ability component, for example, in visual imagery, which is involved in image generation, and motor imagery, which is involved in maintaining an image in one's mind. A different explanation could be the dissociation between body schema, which are online sensory motor representations based on afferent and efferent information, and here represented through the motor imagery tasks, between body structural descriptions, which are described as the visuospatial body maps which are necessary to provide structural descriptions between the relationships among our body parts, here represented through the visual imagery tasks. If you want to find out more about our research, have a look at our poster and I will be at the reception happy to answer any questions. Thank you.